Hello everyone and welcome to our first Q&A webinar of 2024. I'm Diane Stevens, Chief Executive of TOFS. Thank you for joining us this evening. This has proven to be a very popular topic. We've had about 120 bookings, mostly from members here in the UK, but also members in a number of European countries, as well as Australia and Canada, the States, South Africa and New Zealand. So a very warm welcome to you all. Clearly, there's a lot of interest in this lost twin hypothesis that we'll be hearing about shortly. Our speakers this evening ran a popular session at our recent information event at the Institute of Child Health in November past, and they are Dr. Charles Shaw-Smith, whom some of you will know. Charles is consultant clinical geneticist at the Royal Devon and Exeter NHS Trust and one of our long-standing patrons. We're joined also by Dr. Bryn Webb, who's Associate Professor at the University of Wisconsin in the States. A warm welcome to you both. The idea of a lost twin being the cause of OA TOF is something that was first mentioned by one of our trustees, Sue Lewis-Jones, a few years ago now, I think, actually. And some of you contributed your experience at that stage. So I, for one, am really pleased and excited about the developments that have allowed this to come onto our radar again. So we'll hear from Charles and Bryn shortly, and then time permitting, we have some pre-submitted questions that we'll come to afterwards. So if you have additional questions, you feel free to put them in the Q&A chat function. But if possible, if you can listen to the presentation first, as your questions might well be covered during the course of the talk. And we hope to round things up at 8 p.m. or close enough to it. So over to you, Charles and Bryn, if you would like to join us and to make a start on your presentation.
Thanks, Diane, for your kind introduction. It is very exciting. It was first of all a great opportunity to give a talk at the conference in November. I dare say some people who are there are here this evening, and also some new people. And we've actually initiated this project now, and I'll be giving you an update, and Bryn will be following on with an additional talk. So I'm going to share my slides with you. I'm going to aim to talk for maybe 10 or 15 minutes maximum, hand over to Bryn, similar-ish time, and then I hope we will have plenty of time at the end for your questions. So first of all, I want to thank all of you who've contributed so far, to the people who signed up and gave samples at the TOF conference in November, to those who signed up in November and have been patiently awaiting a sample collection pack, which they haven't received yet, but will be receiving, and to those people who've contacted me by email to register interest. And I've had quite a few nice emails and expressions of interest, and I've responded to them, and you will also be given an opportunity to take part. So all people who have expressed an interest will be given that opportunity. Can be a bit slow with research, just to do some expectation management, but we will pursue this, and hopefully we'll get there in the end. And whether a lost twin will prove to be due, whether OA and TOF will prove to be due to a lost twin, that's why we're doing it. We don't really know. We have to be prepared that we're going down a blind alley. I hope not, but that's the nature of science. We make a hypothesis and then we test it. And it can be a bit brutal if your hypothesis is wrong. But we will, of course, feedback the results to you. I'd just like to thank also Sarah and Diane for all their work in organizing this webinar. It's been great. So really, just to say that for most children with OA TOF, and I'm including VACTERL in this as well, but the cause isn't really known. There's a few small percentage causes, such as a chromosome condition, a single gene condition, and so on. But for the most part, we don't know. First, so I'm a geneticist, and I expect things to run in families, but esophageal atresia and TOF usually don't, which is the first slightly unusual thing. So what's all this about twins then? So I've got a few slides about twins. And the first slide I'm going to show is just to distinguish between what are called identical and non-identical twins, because that is relevant. So identical twins result from a single egg and a single sperm, and where you get fertilization. I hope you can see my pointer. I think you probably can. And then at some point after fertilization, the cell divides and you get two new individuals, Sally and Sue. So that's identical twins. And an important point is that the timing of that splitting is highly variable. It can occur very early or it can occur much later. And in fact, if it occurs extremely late, then the end result of that is conjoined twins, which used to be called Siamese twins. In contrast, fraternal twins, or if female, sororal, result from the simultaneous fertilization of two separate eggs by two separate sperms to give two individuals, which may be like-sexed or they may be unlike-sexed, whereas, of course, identical twins are always like-sexed. So I've had a couple of emails, which is why I'm going through this, from one individual who may or may not be on the call. I've just checked. My husband's mother is an identical twin, and his brother has non-identical twins, which is surprising to me as I can't tell the difference between them. And in fact, that is the test for identical or non-identical twins. So obviously, if they're unlike-sexed, a boy and a girl, they're non-identical. If they're like-sexed, the critical question is, can I tell them apart? And if the answer is yes, you can quite easily, then they're probably non-identical. And if you can't, then they're identical. And the other thing that came out of this email exchange that I had was that the parents of the twins had been advised that because there were two separate placentas, then it was likely that they were non-identical twins. But in fact, with identical twins, you can have two separate placentas. If the splitting event that led to the twins happened very early, before implantation in the womb, all a bit technical, I hope you follow that. It's rather an important distinction. We're mainly interested in identical twins in this study, but we will certainly not turn you away if it's a question of non-identical twins. So being a twin is known to be a risk factor for a wide range of congenital, which means the thing is present at birth, and malformation, alteration in the usual structure or shape of bodily organ, which esophageal atresia and TOF fall into that category. And in the, if you like, normal population, a small percentage are twins. But if you look at infants with OA TOF, the proportion who are twins is significantly higher. So that's the first point of interest. The second point that's key, I think, is that what we call the twin concordance rate is very low, which is to say that usually, mostly, one of the twin has the thing, OA TOF in this case, and the other one doesn't. So that's called a discordance. And of course, because identical twins have got identical genetic makeups pretty much, then it doesn't really fit with a genetic cause. So an alteration in a gene or a chromosome, because if it's present in one twin's genes, it's going to be present in the others. So that was one thing that really has made people wonder about what the mechanism is here. Second or third slide about twins, the incidence of identical twins is higher than expected. So most twins are in fact fraternal or sororal, and a relatively small proportion are identical. And I don't have an exact figure, but it's rarer to be identical. And we've all seen that. You see quite a lot of non-identical twins, and it's quite a thing if you see a pair of identical twins. But in OA TOF, children with that, where there is a twin, the majority are like-sexed. In other words, they're both boys or both girls. And that is not what you would expect, because if they were all fraternal or sororal, only about 50% would be like-sexed. So what can we conclude from this? Being an identical twin appears to be a risk factor for OA and TOF. And that kind of seems initially at first sight to suggest a genetic cause, but OA TOF doesn't usually run in families unless it's one of those very unusual genetic causes which account for really a very small proportion of the overall number of individuals with this. And then identical twins are usually discordant for OA-TOF, as I explained. So suggesting that some other mechanism must be at work. So sorry for sensitive computer mouse here, it keeps advancing me. This brings us on to this idea of a lost or vanished twin. So what about a twin lost in early pregnancy? There is some evidence that such early loss is associated with an increased risk of congenital malformations, but not specifically for esophageal atresia and TOF. And I did, through a variety of initiatives, through a survey that Sue Lewis-Jones helped me with, through attempting to put together a case series, even an attempted collaboration with a fetal medicine specialist, I tried to look into this, but always came up against the problem of how on earth are you supposed to know if a pregnancy started off with twins and one of the twins was lost? It's really hard. And you can use surrogates like presence of an empty sac, for example, or was there a heavy bleed early in pregnancy? But these are really hard things to get a handle on. And what I ended up saying to myself was, without any expectation that this plea would be answered, help, I need a blood test that will tell me if an individual who was born as a singleton actually started life in the womb as a twin, a blood test for twins. And in fact, the plea was answered. So these very, very clever people in the Netherlands identified the fact that, and I'm not going to get into technical detail at all about this because it's just too late in the day. But I'll give a brief overview. So what we're talking about here is that the key word is persistent. There is an epigenetic signature, something that you can detect in an individual that and this is lifelong. So even if you're very old, that specific signature will still be there, even though the twin has long gone because the poor other twin was lost in early pregnancy. And this science seems to hold up, and it's really exciting because it means that we can do a DNA test and it will, we hope, tell us if there was a lost twin. And nobody in clinical genetics likes talking about methylation because it's a slightly difficult concept. And the nearest I can get to it is this idea of redaction. So if you think of the DNA sequence as a string of letters and words, it pretty much is that. I often use the analogy of a recipe book. Sometimes, or quite commonly, and in a variety of ways and settings, our DNA, our genetic code, is modified a bit like being painted over. And the effect of this painting over is to almost act like an on-off switch. And it's a little bit like blackening or crossing out words in a piece of text. But the point is that in identical twins, this happens in a very characteristic and identifiable pattern that we can test for, which is absolutely great. So we are carrying out, Bryn and I are really excited to be doing this research study, and we're interested in obtaining DNA samples, which we can collect from saliva or from a swab from the inside of the mouth, from an affected child and both parents. But actually, we don't have to have both parents. We could just have a child or an adult who has a TOF. And this can be OA, esophageal atresia, or TOF, or VACTERL. I think as long as, I mean, you're listening to this talk because there is a TOF in your child or in you. But if there are additional malformations, we're still very interested, so don't be put off. We will ask you to complete a short clinical questionnaire. There's a consent form to sign. We don't need blood, fortunately. Saliva works. One thing I should say, and Bryn may say a bit more about this, we did collect some mouth swabs on the day of the TOFS conference, and I understand from Bryn that preliminary data show that the yield of DNA from those was a bit low. So we may need to come back to you for more. And in fact, one thing that will come up is whether you or your child are able to provide a saliva sample rather than a cheek swab, because saliva tends to give a bit more DNA, but it does require that you spit into a tube, and you need to be above a certain age and degree of coordination to be able to do that. Now, we're not providing individual feedback at this point, although, having said that, if this hypothesis holds, it's something that could be very quickly and easily transferred to NHS genetic clinics. So, but for now, what we will do is analyze the whole cohort, and then we will present the results in a webinar like this, but not with individual feedback. And once we've got to that point, we will explain how you can then go on and get individual feedback, because I know that many of you have a strong feeling that for various reasons, family history of twins, empty sac, early bleed, and so on, that there was a twin there. And we want to help you to get that answer. So I am going to hand over to Bryn now, and I think what we'll do is we'll take all questions at the end of both talks.
So I want to also express that I'm really excited about this study. It's been so nice to be welcomed by this group and to meet up with Charles to explore this hypothesis. So I'm going to say a little bit of what Charles already touched on, but I think, to reemphasize is also good. So the hypothesis here is that persons with TOF or OA may have a specific twin methylation fingerprint. So we're all aware, or most of us are aware, that our DNA is this double helix. That's composed of a sequence of letters like A, C, T, and G, and those letters are a code. So we're not talking here though about the ACTGs or the sequence. What we're talking about is a modification that can be made to the DNA. And so here it's represented as like a methyl group. So it's an actual sort of carbon that can be added to the DNA without changing the sequence. And so this process is really important for a variety of reasons, normal development, and also just a number of processes in our body, which genes to turn on, which genes to turn off, etc. So DNA methylation can be measured with what's called a methylation array. And so here's a picture of an array. This one shown here is made by the company Illumina. And what you can do is you can load 8 samples into this array, and it measures methylation. So this particular array looks at 935,000 sites in our whole genome, and it looks to see if there's that addition of the methyl group or not, and then represents that data. So this is an example of like an array readout, and each column is an individual sample. So there's 7 columns here and 7 columns here. So that's 14 patient samples. And what we're doing again is reading methylation. And so we can see in red, here's the key, that means that sites are more methylated than relatively. And when a site is shown in green, it's a negative number. So that means it's less methylated. And so what we can see, and so each row is at a particular place in the DNA. So at that site of DNA, whether there's methylation or whether there's not, can be read out. So what we can see here is you can see in this first 7 individuals that there's a pattern, where at these locations at the top, the DNA is more methylated. And at these DNA locations at the bottom, the DNA is less methylated. So this pattern we can call a fingerprint. And so similarly over here, you can see the opposite. So here, less methylated, and these other sites more methylated. So you can think of this as 2 different fingerprints that are seen. And so what we're doing with this test is looking to see if a specific fingerprint can be noticed. So there's, as Charles mentioned, so there's a specific methylation fingerprint that's now known to be seen in twins, and that is that Van Dongen paper that was published just in 2021. And so we'll say that fingerprint, for example, is a green fingerprint. And what's important is that this fingerprint is lifelong, as Charles mentioned, and it's seen in all tissues. So we should see it when we're looking at saliva, DNA from saliva. So why it's important is that we can retrospectively diagnose if a person was conceived as an identical twin. So here, when we're looking at a population of people, we can find the green fingerprint and say, okay, that person's positive for having been a twin. So our study design is such that first, a patient consent form needs to be completed. These are a little bit lengthy as far as a research consent. So this study is approved by an institutional review board, this one at the University of Wisconsin. And so it's about 7 or 8 pages that need to be reviewed, but just basically saying information about the study, information that will be isolating DNA, giving permission to provide a saliva sample, etc. Then the next, we'll ask you to complete a questionnaire, a little bit of information as far as the pregnancy, parents' age, if there's been any genetic testing, and then family history. And then we'll ask for a saliva sample, which I'll talk about next. And then we'll perform the array. And then we'll look to see if the individual has the methylation fingerprint for twins or not. So as far as saliva collection for DNA isolation, as Charles mentioned, we did collect some DNA at the conference, and some samples had good quality DNA and other samples had a little bit of lower amounts of DNA with the specific kits that we used. And so that we can make sure that we have enough DNA so that we can sort of replicate results and also maybe use another technology to confirm results, we'd like to ask for some saliva samples that are collected with the specific kit because they give much more DNA. And so the good thing about these saliva samples is that you basically spit into this tube, and then it's stable at room temperature. So it can be mailed back to Charles in the regular mail. It doesn't have to be immediately, if it takes some time to get to Charles, that's okay, because it is stable with the solution that comes with the tube in the package. And there'll be directions more specifically about how to collect it. It's important, not to eat or drink within 30 minutes before getting the sample, etc. And then there's a similar kit that can be for young children that's also by the same company, and it's saliva collection with a sponge. And this one is 60 days stable at room temperature, so it needs to be processed a little bit more quickly, but it should still be a really good option for mailing through the mail, collecting and getting back to the mail and then getting to the isolation. So here what you do with the sponge is you swab sort of at the back of the child's mouth, and then you're not really swabbing the cheek in this one, you're soaking up the saliva. So you put it back in there, you move it 10 times, and then you switch to the other side of the cheek, and then you also move it around 10 times. For this Origin kit with the spit where you spit into, you get quite a bit of DNA. For this swab, in the young children, you get, obviously less DNA. So we might, for the children, ask for 2 tubes to be collected. That would be more important. And then, if we have extra kits, we also might send 2 tubes for the traditional one as well. So as Charles had mentioned, we do want to enroll the person with TOF or OA. and we would like both parents. If there is a known twin, we would like to also enroll that twin. And then it's also we'd also look at fraternal twins as well. So if there's a known fraternal twin, we would definitely like to enroll both twins. So so as far as delivery of results, as Charles was mentioning, when we do a research test, what we like to do is confirm that test in a clinical laboratory so that we can provide a clinical report. That being said, this is such novel sort of testing that really clinical labs at present time don't know, or they don't have experience confirming this type of result. And so until that's been moved forward, or the technology and acceptance has moved forward, it's going to be difficult to give you an actual clinical report for each individual. But that might change over time. And so the main delivery of results will be presentations and webinars to the TOFS community. And also we'd like to publish our findings in scientific manuscripts, and that will also be shared, and we can go over at meetings such as this. So here's a little bit of information about me. I am from Wisconsin. And so if you're wondering where that is on a map, I'm here in Madison, Wisconsin, at the University of Wisconsin. And yes, I'm, very excited to pair up with Charles to do this project.
We've got some questions coming in on the chat, and we have some pre-submitted questions. So perhaps we can run through some of those just now. Just time-wise, we've got another half hour to go through those, if that's okay. So starting off with the pre-submitted questions, we have someone who has emailed us to tell us they have a 6-year-old child born with a weight TOF, and then they went on to have twins who are now 3. Could there be some connection? Is that question?
Okay, thank you for that. I would say it's certainly possible. The key question, of course, is whether or not the affected child was a twin, and that a twin very early in pregnancy. And that's the question that we're trying to answer. I'd be interested to know if the set of twins that you had were identical or non-identical. If they were identical, that would be interesting. It's less clear whether there could be a link with non-identical twins, but we don't really know. But the hypothesis we're primarily testing is regarding identical twins. And there may be some other family history of twins as well. So I've certainly had emails from people where there are several sets of twins in the family, which really does make me wonder.
Mm-hmm. So we possibly need more information from that particular member then, Charles, at some stage. I
think I would say please register for the study, and we'll try and answer the question.
Yeah. And the other thing I should mention is that the email address for both of you will be given out at the end of this presentation as well. It's available on the TOFS website, and we'll give it out in the chat at the end of the presentation. So thank you for that, Charles. Our second question is from someone who tells us they've requested a genetic study for their son. He's gene 11 with some missing information, but they couldn't relate it to anything.
If the person who submitted that question has got a letter with a bit more detail in it, I'd be very happy to take a look at it by email, but it's not really enough information to answer in a forum like this.
Okay, I understand. And that brings me on to a question we got in the chat, actually, from someone who joined a bit late and missed the introduction. And they're interested in the possible causes, give an explanation for a weight TOF. They've recently had genetic confirmation that their daughter has 22q deletion.
Okay.
Would this rule out lost twin hypothesis for them?
Yes, I think it does. So if you think back to my original slide, there's a small proportion of children who already have a known cause for their OA or TOF. And there's a handful of genetic syndromes which include Down syndrome, actually, 22q microdeletion syndrome, and some other rarer syndromes. Feingold syndrome is another one. And I think what I would say is that if your child has been given a diagnosis of a specific genetic syndrome by a clinical geneticist, then we feel that it's already explained, and we would not be looking for a further explanation along the lines of a lost twin. But that is a great minority of children who are affected.
Thanks, Charles. Third question is, if there was no indication that a second baby was miscarried early in the pregnancy, is it still possible that it was a lost twin?
Yeah, I think I'm sure that it's entirely possible that there could be a lost twin without any external signs such as bleeding or pain or whatever. I think the answer is we don't really know.
Right.
It would be, leaving aside OA and TOF, it would be quite interesting just to get a general answer to that question about how many children are conceived as identical twins and then one of the twin pair is lost. Because of course, we're saying that we think that the early loss of a twin may cause esophageal atresia, but if that is the case, we don't know what proportion of early lost twins give rise to a child with esophageal atresia. It might be a high proportion or a low proportion, and we just don't know.
Okay. Can this happen to a mother who isn't a twin, but their parent is?
Yeah, of course. So the key in our hypothesis, the key question is whether your child was conceived or had a twinning event early in pregnancy. Whether or not the mother is a twin is kind of not really material, although it adds a bit of weight if there's a family history of twinning, which could be on the maternal side or the paternal side.
My sister had a very heavy bleed when 7 weeks pregnant, and her baby had a 2-vessel umbilical cord. Is this indicative of a twin?
I'm assuming the baby had a TOF as well, or maybe not.
We don't have any more information, I'm afraid, with this, but I assume so.
I think there's not enough information there to say that there was a twin, actually. And one thing I learned from Baski, the fetal, I can't say his name is so long, I can't even remember how to say it. He's a lovely guy, though, from Sri Lanka. He's a fetal medicine specialist in St. George's. And he put me very straight on bleeding in early pregnancy. He said it's very common, very common, maybe up to 50% of pregnancies. So to say that, some of them will be miscarriages, some of them will be lost twins, some of them will be just a bleed. But to say that a bleed is indicative of a lost twin is a stretch. We can't say that. And that's why this test is going to be much more specific and help us to answer the question properly.
Fair enough. Another question, Charles, is there any way to help provide data if parents are deceased or live in the opposite end of the world?
Yeah, we can just recruit the affected individual without the parents. And we as Bryn said earlier, we're happy to do that.
I'll add, it might be possible to collect internationally as well. So we can look into that for the parents.
Yeah, so we can certainly look into that. Yeah.
This is another one about a bleed early in pregnancy. So someone had a bleed at around 10 weeks pregnant. By my 12-week scan, the bleed had been reabsorbed. Is it likely that that was a lost twin?
That's quite late, actually. And I think one of the points I made in my talk in November was it's amazing how early in pregnancy the esophagus and trachea are formed. And by the time you get to 10 weeks pregnancy, it's all happened. So losing a twin at that point can't sort of go back and cause it. They've already formed. So we're more interested in, the esophagus and trachea are formed between about, 3 weeks and 6 or 7 weeks, something like that. So it has the event, whatever the event is, it has to happen in that time frame.
Sure. Okay. I think you might have responded to this, but let's ask it anyway. Does the same hypothesis apply to VACTERL, or is it just a way to? Absolutely. Probably even more so. We love to recruit individuals with VACTERL while sympathizing with having more malformations to contend with. Sure. This person had a bleed early pregnancy. What the chance there was another baby, i.e. lost twins. So obviously, hopefully they will join the study.
Yeah.
And okay. Is VACTERL also considered, so that's the same one, let's not do that, the same one again. Does it
Yeah.
have to be identical twins? I was told at an empty sac, a 12-week scan suggesting not identical twins. Again, you've sort of answered this, but maybe
Yeah.
you'd respond.
An empty sac is consistent with identical twins. It doesn't rule them out for the reason I was talking about. If there's a very early splitting of the embryo and they implant in two separate parts of the womb, you're going to have two sacs, but they could still be identical twins. Okay.
Yeah.
It's a slightly tricky, actually this whole twinning thing causes no end of confusion amongst medical professionals. And I'm still learning about it myself. I mean, it is a fascinating area of medicine, but it does get a bit confusing at times. But just to emphasize again, an empty sac could be identical twins, and it could be relevant.
Okay. Listen, interesting one. Could losing a twin add to my daughter's high anxiety of not wanting to be alone? She's showing signs of codependency.
Yeah, I mean, that's a fascinating question. I think it's very hard to say that would be a possibility, simply because we're talking about a stage in the development of the embryo when there is no nervous system whatsoever. So, if you like, where's a lost twin at a very early stage, it's that there can't be any, if you like, memory of that in the surviving embryo. So it's an interesting thought, but and I'm probably the best qualified person to answer it, but I think it'd be a stretch. I don't know if you got any thoughts on that, Bryn.
Yeah, I think we don't have enough information to address.
We don't have enough information, no.
But I agree,
Sure.
It's very interesting question.
I think let's go to some questions that not respond to. Some of these might be duplicating what we have already asked, but if you don't mind, if we can run through them fairly quickly, there's 13 of them. So if I'm duplicating stuff, please tell me and we can move on. So this person would like to join. She has twins. Only one of them is away. Could he join the study?
Yes, that's what we're looking for.
Ideally. Okay, that's
interested to know if they're identical or non-identical twins.
So maybe we'll get a response to that.
Are the genetic markers?
So
genetic markers on fraternal twins that don't appear to be from the study, that the Dutch study. This applied to monozygous or identical twins.
Okay, thank you. There's not a question on this one. My husband's sister has had have a question.
So having tendency to have twins can definitely run in families, and it just strengthens my idea or our idea that there could be more twins at the beginning, but one of them was lost.
So again, join the study, I guess.
Definitely join the study.
That's the response for you. Liz is asking, will you be sending out the information to join the study? All of the information is on our website.
But you have to get in touch. Yeah, get in touch either through TOFS, or you can email myself or Bryn directly, and we will send you out what you need to join the study.
Okay. Kirsty asked, my TOFS father is an identical twin but having a twin parent link. Again, very similar to what you've responded.
Definitely could.
So hopefully that's your answer, Kirsty. And Charlotte tells us that her 4-year-old was born with VACTERL. She had an early scan at 6 weeks but showed no sign of a twin. If there was one, could it have been absorbed at that stage?
Yes, it's possible. Yeah.
Is it likely? Is it, when you say it's possible, Charles, even at 6 weeks?
Well, yes, because this I don't really like the word splitting, but you know what I mean. When the embryo can divide in two at a very,
Right.
very early stage, okay,
Okay.
any time from a few days old, and we don't know what if this hypothesis is correct. The next question will be, what is the critical time for the splitting event to occur? Is it early? Is it middle? Is it late? But if it's early, there may absolutely not be any sign whatsoever of a twin at a 6-week scan. It's all history by that point.
Okay, I understand. Okay, hopefully that answers your question, Charlotte. Moving on now to Alex's question, which I think might be more for you, Bryn. We might be able to give Charles a rest with this one.
Yeah, I've been answering questions in the chat.
Yeah. So this question is from Alex. What other congenital disorders have no known genetic cause but have a twin correlation? Are there any?
So so we think of some of these as recurring constellations of embryonic malformations, and so that's sort of a name for ones that seem to run more commonly in twins. And so one of those is actually Mobius syndrome, where kids have inability to smile. There's other ones as well, all rare-ish ones. So I would say esophageal atresia, tracheoesophageal fistula with VACTERL is one of the commoner ones. Another one is oculoauriculovertebral spectrum, also called Goldenhar syndrome, where the ear doesn't form as well and the jaw can be smaller. But basically there's a series of conditions where they keep having sort of the same symptoms or a constellation of features that are seen more often in twins, that we wonder about the same sort of processes.
Thanks, Bryn. Have you anything, Charles, to add to that one?
No, that's fine.
Could this be part of a wider study? Alex asked as well. And he also asks.
Yeah, I know other people are looking at this with other conditions, but we're because I've got this. I've been interested in this in TOF for a long time, and I'm not about to change that interest now. So I'm sticking with TOF. Thank you very much.
Thank you. Chana asked, is the hypothesis that the loss causes it, or just that it is more prevalent in twins and therefore also in lost twin?
That is a great question. I had to really think about it. I've been reading it. So the hypothesis, as I understand it, is that the splitting event of the embryo in some way leads to the development of a congenital malformation. So it's actually not relevant to whether or not the child develops a TOF, whether or not the other twin survives, if you follow me. It all sounds a bit horrible, this. I'm sorry. And I know twins can be a very sensitive subject, by the way.
Yeah.
You know, the idea of a lost twin is not without emotion, and I am aware of that. But nevertheless, you know, we're trying to understand why children are born with TOF, and this is the hypothesis that we're testing. So it's not the fact that the twin is lost; it's the fact that there was a splitting event in the first place. I think. I don't know if you want to add to that, Bryn.
Sorry, I was answering the chat, and have
you written something?
Yeah, I've been answering some of these questions in the chat.
Okay, fine, great.
Okay, well, listen, shall we move on, because we have quite a lot of questions to do, and we're sort of getting through it about 10 to 7. So sorry for rushing through these a little bit, but it would be good to try and get through as many of them as we possibly can.
Yeah, let's keep going.
So some of them are. Judy tells us she's going to try and find the information, or some may remember. We have one from Carly. What are we saying for those who do have identical children? She has triplets, 2 of which are identical. Are you saying there could have been another identical baby that was lost, or are we just saying the presence of an identical twin is a factor?
Yeah, I don't know enough about triplets to give a sensible answer to that question. So I'm sorry, I'm going to duck it, but I will think about it. I guess it's just possible that you have, with triplets, you have you conceived two fraternal twins, but both of them underwent a splitting event to lead to identical twins. So you started off with quadruplets, and then one of them was lost. That is a hypothesis. But that's a hypothesis that would be consistent with what we're saying. Sorry, Bryn.
I did answer a question like that. So if they were identical, though, triplets, we would see that, the signature as well. But yeah, Charles is going to, like, there's different possibilities to get triplets.
Yeah.
Thank you. Someone, I hope we haven't done this question before, because I'm losing track now about what we've done and what we haven't done. So someone attended an early scan around 6 to 7 weeks, and it's because she had bleeding, and she was told at that stage that a lost twin was suspected as there was evidence left. Do you believe in the lost twin having a strawberry birthmark? This is something we found on the internet, and our little TOF has a strawberry mark.
I've not heard that, and what I would say is, let's use the best method that we have to try to find out if there's a lost twin, which I would say this is the only kind of a methylation test is the definitive test. I don't think a strawberry birthmark would help.
Right. Okay. Thank you. And then we have Judy. So Judy, I'll come back to that one in a little bit, because I think you're asking about a different study that I'm not sure that Charles was involved in, actually. So I'll come back to that question. Joanna's responding. That was the question we asked earlier, and you had said, were they identical? They were non-identical, she's telling us.
Okay.
Okay.
Yeah.
Liz is looking some contact into. We'll send that out. Charlotte is saying that her understanding is that only fraternal sororal twins run in families, and that identical twins occur randomly. Do you know if that's correct?
Well, I would say no. Identical twinning can run in families as well.
Okay.
Although it is rarer, but it's certainly reported.
Okay. All right, and this is a follow-on question from the lady who asked about confirmed diagnosis of the 22q deletion. And Catherine's asking, is there any chance that the results could be false?
What?
So that's related to the recent testing they've had.
Okay.
She had concerns as to how the bloods were taken for that test, and sorry to interrupt you. Our daughter so far has no other symptom signs of 22q deletion apart from away. Yeah.
Well, what I would say to you is, if you have any concern at all that there could have been an incorrect result, and this comes up with some of my patients occasionally, and I always say the same thing, which is, let's repeat the test on a new sample. And that's the only way to be definitely sure. Otherwise, you'll never get rid of that nagging doubt. So that's what I suggest you do. And if you make an approach to the genetics team who tested your child, then they should agree to that.
Okay. Question now from Una. Does the lost twin hypothesis correlate with vascular causes for some of the features of VACTERL?
Some of the... Well, I'm not sure what the vascular causes are. There are hypotheses out there that some children are born with congenital malformations because of some vascular issue. So like a bleed or a thrombosis or some event in early pregnancy that interferes with the development of the child. And that's certainly been postulated for VACTERL, but it's impossible to get proper evidence for that. So it really is only a hypothesis, and it's very difficult to test. Whereas the lost twin one, we now can test. So that's where we're putting our money, if you like.
Okay. Thanks, Charles. Claire is asking if you could explain the splitting event again, just briefly. Do you mean when the embryo splits into two to give identical twins, or the event when the twin is lost?
Yeah. So, okay, so you start off with a fertilized egg. So that's a single cell. And then it divides a few times and you get a little ball of cells. Then that ball of cells splits in two. Okay, so you now have two genetically identical embryos. And then they carry it on and they will implant in the womb if they have not already done so. And then, so that's, if you like, that's event number one, the splitting. And at some subsequent point, one of those embryos is lost. Okay, so we're not saying that the lost twin relates to the splitting event, because if that was the case, then no identical twins would ever be born. But of course, they are born all the time. It's just that in some cases, one of those twins is not viable, and we have no idea why that is, but that is what happens. So I would call those two separate things, and I hope that's a reasonably clear explanation.
Thank you, Charles. Right, I'm just going over these to see which ones we might have missed. Anyone asking about sending out information to join the study, please email Bryn and Charles. Their email addresses will be on the website, and Sarah's going to pop it in the chat as well before we actually finish the call, so that you have them and you can take a note of them.
I just want to answer, Diane, I just want to answer the last question and clear that up. So, and it kind of relates to my previous comment. So it's a good question. If there is a TOF who has an identical twin, both alive, would this go against the TOF hypothesis? No, because it's not the losing of the twin that we think is the event that led to the TOF. It's something to do with the way the embryo splits. So I would say that the presence, and we know that's why I was talking about the twin concordance rate. We know that it's often or usually the case that there's a twin one has got the TOF and twin two doesn't, but it doesn't invalidate the hypothesis.
And we'd be very interested in having individuals in that situation participate.
Yeah, we would. Please join the study. It'd be great, because if you like, you would be serving as a positive control.
Mm-hmm.
You would be saying, we in this case, we actually know that you're an identical twin, so we can use that to kind of validate our testing method.
Okay. We've had another question from someone in Canada. Is participating an option? My husband and son both have OA12, but so far no link between them.
Yes.
Yeah, I would recommend seeing a clinical geneticist, because of just to rule out that condition called Feingold syndrome.
Yeah.
Which is one of the very unusual scenarios where OA12 can run in family, and I imagine that has been ruled out. And we know that, it's possible to have a second occurrence in the family by chance, because if you think about it, TOFs occur 1 every 3,000 pregnancies. So in 3,000 squared is 9 million. So in 1 in 9 million families, there'll be 2 TOFs that have happened purely by chance, but that's very unlikely, isn't it?
Sounds very unlikely to me anyway, with those numbers. But anyway, so just a final one here. Sarah has said that her daughter also has tetralogy of Fallot, which she's heard of several 12 away children having.
Yeah.
And she comments it's a very exciting study, so she's looking forward to taking part.
So that's venturing into the VACTERL spectrum territory. And yes, we'd be very keen to have your involvement in the study.
Okay. And Vicky has commented, just a comment about the psychology of lost twin. It's not exactly mainstream, but there are definitely people working with this. And she quotes the study Twin Dynamics and the Baby's Experience, a prenatal and perinatal somatic approach.
Yeah.
And there's a link to an article which people can follow if they're interested in reading more.
Thanks, Vicky. Nice to see you on the call. Vicky and I go back a long way. I hope you join the study.
Okay, Judah, you had asked, I have lost your original question, but you had asked about a different study that we had, that some other people had done, had been involved in, I think a few years ago, that you were hoping to get some information from. So I'll drop you an email about that. I haven't heard anything recently in relation to that. I know there was some information published, but it's a separate study. So I hope I've remembered your question properly, because it's disappeared off my list of questions. And please, if I have missed one, can you drop your question in the chat again? I know we're almost out of time. But I think we've covered all of those. There's a couple of questions just from that were pre-submitted that we didn't cover. Because I thought we'd sort of touched on subjects very like it. Does heavy bleeding during early pregnancy have anything to do with this?
Possibly.
Could be an indicator. At how many weeks would a lost twin be miscarried? Is there any way to know if you had a lost twin?
That's what we're trying to find out.
Take part in the study? Yeah. Why is it more common for twins? Is it one-egg twins specifically? Is the final
One-egg
question.
twins, yeah. So I think that's the hypothesis, that so one-egg twin is the identical twin, and the two-egg twin would be a fraternal or soror.
Okay, thank you, Sarah.
Yeah.
Perhaps you can tell me if
I'd
I've
love it if
missed
we knew the
anything.
answer to that question.
Yeah, that would be good. I think I'll just
We'll
down to the
let you know
bottom.
as soon as we've got results.
Yes, and that's responding to Magella, who wants to know
Yeah.
when will they be published, finalised, and I guess we don't have a timescale for that at this stage.
No, this is research. You never have a time scale, unfortunately.
Okay, well, that's great. Sarah's going to put the details of those email addresses, if you don't mind, Sarah, into the chat. Yes, you're going to put them in the Q&A. I think you're hoping to do that, Sarah. But anyway, I think we better say that was really informative, and thank you for being so patient with all our questions. And also thanks to everyone who has attended tonight. We will make a recording available of this video within a few weeks, and if you're watching the recording afterwards on YouTube, please like the video and subscribe to our channel.